morning. Today we will discuss about the biofertilizer, their classification, what is the use of biofertilizer, what is the scope in the crop productivity and increase its soil productivity. So in this class we have discussed about all these things about the biofertilizer, advantages, disadvantages, how it is differ from the inorganic fertilizer. So students, first we start from the what is the biofertilizer. As you know, as per their nomenclature, biofertilizer, bio means life, जिसमें जान होता है, जीवित रहता है, living thing, and fertilizer, that is a plant nutrient. Those materials containing living cells of microorganisms which is capable to fix the atmospheric nitrogen. As you know, in the atmosphere about 79% free nitrogen is available, that is directly not used by the plants. So through this microorganism, nitrogen fixing microorganisms, phosphate solubilizing bacteria, for potassium and zinc solubilizer bacteria, you can assess, plant can assess the nutrient from the atmosphere as well as fixed form in the soil. So on the basis of these things, this biofertilizer is a defined like it is a carrier of liquid based microbial inoculants. It has containing living cells of efficient strain of the microbes which is increase the crop productivity, enhancing the soil fertility and fixes atmospheric nitrogen as well as solubilize the phosphorus and potassium, that is fixed form in the soil. As you know, in the acid soil, phosphorus fix in the form of calcium phosphate, whereas the availability of the phosphorus use efficiency in the soil is only 15 to 20%. But through this biofertilizer, you can solubilize and plant can use phosphorus up to 25 to 30%. So after the definition, you can classify mainly two part. First is symbiotic nitrogen fixation and second is a non-symbiotic nitrogen fixation. Symbiotic means relationship, जैसे आप और हम दोनों मिल करके किसी काम को करें और दोनों उसका फायदा उठाएं. नॉन सिम्बियोटिक मीन्स आप अपना कमाइए खाइए, मैं अपना कमाऊँ खाऊंगा. Similar thing with the plants, that nitrogen fixing bacteria within their tissues, the best studies is an example in the association between legume and bacteria in the genus like Rhizobium, Azotobacter and Azospirillum. So symbiotic relationship, both mutual understanding, plant will provide the host of the bacteria and bacteria will provide the nutrient to the plant. Second one is non-symbiotic bacteria. Some bacteria is available in the atmosphere that is not any relation with the plant. They are independently work for the nitrogen fixation, especially Azotobacter and Azospirillum. So it's relatively non-creative molecule that is metabolically useless to all about the few microorganisms. Looser non-symbiotic relationship between diazotroph and plant are often referred to as associative, as seen as nitrogen fixation of rice, not only rice, that is for also for the grain legumes like maize, ragi, wheat, mustard, that is also upland crop. So as well as rice is concerned, rice is mainly suitable microorganism is the azolla and blue-green algae. We will discuss later on. You can see here, this photograph shows how microbes enter in the root cortex through the root hairs and how it fixes atmospheric nitrogen. What happened, how you can say the nodule, type of nodule. You can see here the first slide. This is the root hair. You can see here the microbial bacteria of the Rhizobium enter in the root cortex from the one root threads, and you can see the slide B that is enter in the root cortex, and cortex is swelled when this multiply like 2 to 4, 4 to 8, 8 to 16, 16 to 32, and its root cortex and cuticle layer is swelled like a eggs and look like a nodule. That's why we are saying this is the root nodule. So first step to third step, starting from the root hairs to the develop in the nodule form. And if you uprooted the any pulses crop like maize, like chickpea, pea, pigeon pea, any pulses crop, you can see the root nodule in the root zone. Look like a pink color. If pink color is not available, that root started degenerated. So better root nodule is like a pink color. Pink color means active bacteria is available in the inside of the root.
Sure.
Then come to the classification of the biofertilizer. First one is a symbiotic and then non-symbiotic. So comes under the example of the symbiotic and non-symbiotic both. So first one is a nitrogen fixing microorganism. That is, that bacteria is capable to fix the atmospheric nitrogen. Second one is phosphate solubilizer. Some bacteria having capacity to solubilize the fixed phosphorus, fixed in the form of calcium present in the soil. Second one is also solubilize the phosphate and micronutrient mobilizing microorganism. So these three type of microbes is very beneficial for the crop production, not only for the crop production as well as increase the soil fertility and give the sustainable production of the agricultural crops. So come to the first, nitrogen fixing microorganism. First one is rhizobium. It is a very clear cut nomenclature. Rhizobium means ऐसा बैक्टीरिया जो रूट में रहता हो, जड़ के साथ रहता हो. And rhizobium culture is suitable for the all leguminous crops as well as some oilseed crops like soybean and groundnut. Second one is Azotobacter, that is non-leguminous crop for mustard, wheat, maize, millets, etc. Third one is Azoospermillum. It is for the especially for the millet crops like madua, gundli, etc. And as well as suitable for the upland rice, where you are producing direct seeded crops. So it is suitable for the upland rice as well as millet crops. Next one is a blue-green algae. It is suitable for the submerged condition of rice. Submerged means जहाँ हम transplanting करते हैं उस rice के लिए ये better है. But Azoospermillum is not better for the transplanted rice because Azoospermillum is aerobic bacteria. It's need oxygen for their survival and their multiplication. But in case of blue-green algae, you can use in the submerged condition because submerged condition of the rice is provide the almost all good condition for their growth and development. As well as azolla, it is also beneficial for the transplanted rice. So BZ and azolla both are very suitable for upland rice, transplanted rice for nitrogen fixation. These are the some biofertilizers that we are in our university Department of Soil Science and Agriculture Chemistry also producing, like rhizobium culture, Azotobacter culture, Azoospermillum for millet crop, then blue-green algae for lowland rice. PSP culture, as you know, in the acid soil there is a very problematic for the phosphorus utilization, and most of the phosphorus are fixed in the soil. So this PSP culture is very useful to solubilize the fixed phosphorus and provide to the plants. Then vermicompost, it is a type of organic manure. Then AM culture, that is stand for arbuscular mycorrhiza. It is suitable for phosphate solubilization as well as micronutrient mobilizer. Then fungal culture, this culture is very suitable for lignin decomposer or cellulose decomposer for rapid decomposition. So these are the biofertilizer we are producing in our department for the farmers' use. First, then come to the biofertilizer, rhizobium culture. What is the rhizobium culture? It is a soil bacterium it produces root nodule in a leguminous crop as well as some oilseed crops. Some oilseed, not all crops, like soybean and groundnut only. It is a symbiotic association with root nodule. It fixes atmospheric nitrogen about 45 to 60 kg per hectare as per literature, but on an average in the farmer's field, you can save 40 to 45% nitrogenous fertilizer. So in the practice, 40-45% is a very good example for it is on an average fixation in the farmer's field, but in the literature it is fixes about 45 kg per hectare to 350 kg per hectare by the some leguminous plant like alfalfa and etc. So it increase crop yield by 15 to 20% by the use of this rhizobium culture, even in the pulses, even in the oilseed crop, even in the cereals. You can increase through this biofertilizer 15 to 20% crop yield. It is a very important. इसके प्रयोग से न केवल नाइट्रोजन रासायनिक उर्वरक की बचत होती है, बल्कि इसके अलावा आप इसको 15 से 20% क्रॉप के यील्ड को बढ़ा भी सकते हैं. Then come to the Azotobacter. Azotobacter, it is a type of very important non-symbiotic microorganism. There is a two types, symbiotic and non-symbiotic. This is the example of the non-symbiotic microorganism. It fixes about 35 to 45 kg nitrogen per hectare. As you know, this in the atmosphere about 79-80% nitrogen present in the free form that is useless for the plant, but through this Azotobacter microbial inoculants you can save 30 to 35 kg nitrogen per hectare. It increase 10 to 20 kg, 10 to 20% yield in cereals. Cereals means maize, millets, sorghum, then also mustard, etc. Then 10 to 15% in vegetable. As you know, Jharkhand is a vegetable growing state, and through this Azotobacter we increase about 10 to 25% vegetable yield through this Azotobacter culture. It also secretes some plant growth hormones. It is a very good character, very important character of this Azotobacter. Rhizobium only provide the nitrogen, but Azotobacter not only provide the nitrogen but also secretes some plant growth hormones that is very useful for the plant growth and plant production, also increase the crop yield. It can be used for non-leguminous crop like wheat, maize, mustard, vegetables, especially potato, tomato and etc. Other vegetable crops. So this is a very important for cereals and vegetables also. Then come to the Azospirillum culture. Next microbial inoculants is Azospirillum culture. It is a very common soil and root bacterium. It is found in the soil around plant roots and root surface, and it works free, not like a rhizobium culture. Rhizobium culture working with the symbiotic relation. प्लांट और बैक्टीरिया दोनों मिलके नाइट्रोजन फिक्सेशन का काम करते हैं. एक रहने के लिए घर देता है, दूसरा उसको खाने के लिए न्यूट्रिएंट प्रोवाइड करता है. बट हियर केस इज डिफरेंट. इट इज अ फ्री लिविंग माइक्रोऑर्गेनिज्म वर्क फॉर द नाइट्रोजन फिक्सेशन and fixes about 30 to 35 kg of nitrogen per hectare in the soil. 30 to 35 kg, as more or less like a Azotobacter. Then it's increase rootlet density. What happens? If increase the plant rootlet density, then what happens? The nutrient absorption is more, moisture absorption is more. So it is a very good character and very good role of this Azospirillum to increase the rootlet density. Means root को बहुत ज़्यादा डेवलप करता है, सॉइल में फैलाता है. जितना ही एरिया में रूट डेवलपमेंट होगा उतना ही जो है प्लांट का न्यूट्रिएंट का एब्जॉर्प्शन ज़्यादा होगा. फिर क्या होगा? क्रॉप ग्रोथ बढ़ेगा. देन रूट ब्रांचिंग आल्सो रिजल्टिंग इन इंक्रीज द uptake of the nutrient. It may be for the nitrogen uptake, phosphorus uptake, potassium uptake and micronutrient uptake. So all are increased through this use of this Azospirillum culture. It increases crop yield by 15 to 20%. Again, by the use of azoospermillum culture, farmers can increase their crop yield, that is 15 to 20%. It is suitable for the jowar. It is used in the jowar, bajra, maize, ragi, small millets and forage grasses. आपने देखा होगा एटमॉस्फेयर में जहाँ भी कहीं घास लगा हुआ है बिना किसी यूरिया के डाले, हम यूरिया नहीं डालते हैं फिर भी वो ग्रीन रहता है. ऐसा क्यों होता है? ऐसा होता है ड्यू टू दिस माइक्रो ऑर्गेनिज्म. एजोस्पिरलम इज कैपेबल टू फिक्स द एटमॉस्फेरिक नाइट्रोजन एंड वर्क एज ए नाइट्रोजन एज ए यूरिया. बिना यूरिया डाले आप इसको यूज कर सकते हैं. You can see here, this is the very good example and isolated by myself, Azoospermillum brasilensis. We can cut the root and pour in the culture media. Culture media is suitable for the growth of the Azoospermillum. And you can see here we put up this root cut, and this root cut give this azoospermillum bacteria, and it is a microaerophilic, means जिसको ऑक्सीजन थोड़ा सा चाहिए, ज़्यादा नहीं चाहिए. दैट इज़ हार्मफुल फॉर द प्लांट, लेकिन अगर कम मिलेगा तो भी ये डिस्ट्रॉय हो जाएगा. सो रूट को डालने के बाद इट्स इंक्रीज आउटर लेयर, दैट्स वाई इट्स नोन एज microaerophilic. Similarly, in second slide you can see here this azoospermillum culture is hang upper layer of the medium. That's its requirement fulfilled by the atmosphere or present in the air. Come to the what is the use, how to use this biofertilizer with the seed. So there is a three way using for the nitrogen fixer, phosphate solubilizer. Like first one is, that is three type, that is seed treatment. First one is seed treatment, second one is root dipping, and third one is soil application. Seed treatment is very suitable and very economic, but soil application is more suitable but not economic. So we prefer seed treatment and root dipping. Root dipping, it is especially for the crop which is suitable for the nursery transplanted crop like rice, vegetable crop like chili, tomato, etc. जिसका सीड बहुत छोटा होता है, स्मॉल साइज ऑफ सीड इज नॉट सुटेबल फॉर द सीड इनोकुलेशन, बट बिग साइज लाइक सोयाबीन, pea and soybean and rajma and etc. is suitable for the seed treatment. But for the vegetable crops, root dipping is very useful and important for increase the crop production. These are the systematic diagram how to multiply the microbial inoculants if you go for the multiplication or production of the biofertilizer. What will the steps involve for this multiplication? First is you isolate the root nodule from the leguminous plant or oilseed plants or any other plants. Crush the root nodule in the petri dish and transfer in the test tube with the slightly either bacterial culture or in distilled water. Then prepare a broth culture inoculation having a medium, artificial medium that is suitable for the rhizobium, Azotobacter or azoospermillum. That is a similar process for the rhizobium, Azotobacter and azoospermillum as well as PSB. Then transfer to the other flask and prepare as a primary inoculum. Then transfer to the shaker, that is for the 72 hours it needs continuous shaking for 72 hours, because during this 72 hour the multiplication rate is very high, and it needs rotary shaker because rotary shaker is capable to create a vacuum in the flask. That's why the oxygen, filtered oxygen come from the atmosphere, and microbes use as a nutrient, as a energy. Then after you transfer in the big culture or Hopkins culture bottle and also shake for the 72 hour. It may be rotary shaker or it may be manually, and then after full growth of these microbes, then transfer to the some carrier material. Carrier material may be you can use as a lignite, as a compost, as a press mud, as a fly ash also. So as per availability and suitability, and as per analysis of the different liming, different carrier material, lignite has been found very efficient and economic for this multiplication of this carrier material, microbial inoculants. You can see here, this is the laminar flow. This instrument is used for the transfer, aseptically transfer of the microbes, like one flask to another flask. If you start the multiplication or production of the biofertilizer, this instrument needed for aseptically transfer of the culture media, inoculation, during inoculation, or any other experimental research. This is the rotary shaker. You can see here, this is the scene of our laboratory. This is the rotary shaker, and it's rotate about 22 hours for their full multiplication for production of the biofertilizer. This is the Hopkins culture bottle. This is suitable for manual multiplication of the microbial inoculants. It may be Rhizobium, Astrobacter, Azospirillum and PSB. So you can, this is the capacity of the 4-liter culture bottle. You fill up about 3/4, 1/4 is vacuum for the oxygen. That oxygen filtered through this cotton and enter in this flask, and bacteria and microbes are utilized. This is the horizontal autoclave. This instrument is used for sterilization of media as well as carrier material. Sterilization means you can remove the living things or any living material, organism free from any living things, and you can say it is a मतलब डेड कर देना पूरे मटेरियल को, चाहे वो लिक्विड फॉर्म में हो या कैरियर फॉर्म में हो, दोनों को डेड करना, स्टलाइज करना, ये इंस्ट्रूमेंट उसके लिए बहुत ही यूजफुल है. दिस इज अ fermenter. It is used for the commercial production of the biofertilizer. If you are not commercial, then go for the Hopkins culture bottle, and manually you can multiply the bacterial culture. But you, if you want for this commercial production of the cultures of the microbial inoculants, you must involve this automatic fermenter. It provides the aseptic condition of the microbes, provide the desired oxygen, maintain the pH, and reduce the contamination chances. As well as in the manual multiplication, contamination charges is more than this fermenter. This is the process of the mixing. After manual multiplication of the microbes, you can mix by the hand with the protected condition and protected room that is sliced by the ultraviolet rays. These are the method of the seed inoculation. Earlier we discussed about seed inoculation. You can see here. Here this is the plain seed. This is the Rhizobium culture. After coating this seed through the Rhizobium culture, it's look like this black seed. Then if you need, your soil is acidic, then go for the plating of the priming of the lime. So after second layer of the liming, this look like a white seed, just like a white A. So this is the process of the seed inoculation. You can see here it is an untreated plant. I want to communicate the message how biofertilizer is a very important, and what happened if you apply and if you not apply. If you not apply, this is the untreated. हमने कोई Rhizobium culture नहीं डाला. You can count the root nodule. How much? 10 or 12 maximum. But after inoculation of the biofertilizer, especially Rhizobium, you can see like this structure. You can't able to count these nodes. So number of bacteria more enter in the root cortex, and manufacture or made the more root nodule, so that the nitrogen, if root nodule is more, then nitrogen fixation is more. So after inoculation, you can see this type of root structure, especially in pulses and some oilseed crops. This is the example of Rhizobium culture in the chickpea. You can see how much nodule is present after inoculation of the Rhizobium culture.